All right, I am Mr. Ferguson, and this is the DNA replication interview. So why do we need to replicate DNA? Well, in short, it's just because you need to make new cells due to damaged cells or the concept of meiosis. But it's to make new cells. Now, remember from one of the previous interviews, not all tissues, therefore not all cells, go through replication. Cells that go through replication are involved with tissues, epithelial tissue or connective tissue, and the other two tissues don't replicate. Now, as we go through these, you're going to see some interesting shapes. If you remember, it's been stated earlier that enzymes they're proteins, and they are protein blobs, and they're just generically shaped. So we got some interesting shapes. They're going to represent enzymes. And that's just to help with kind of imagination and following along. Now, there are far more enzymes than what we're actually going over. It depends on which textbook you read. You can categorize them a little differently, and many enzymes work together in conjunction. But we're just kind of going to break this down into seven enzymes and what they do to help the process of DNA replication. So, we got 3 different steps for DNA replication. We got initiation, elongation, and termination. The first step, initiation, is going to involve 3 different structures, enzyme-like structures that are involved with replicating DNA. And that is this one right here. So this is DNA helicase. This is the one that kind of gets it started. So you know DNA is double-stranded, it's wound up together. DNA helicase comes in and starts the unzipping of DNA. This creates what's called the replication fork, and this is located right here. Now you got to keep in mind there's another DNA helicase that is going the other way down this strand. So this is just randomly done throughout the DNA. It's not done at the ends of the DNA. It's within the DNA itself. And so it creates this bubble that actually forms. And we call this the replication bubble because you got one helicase going one way and you got another helicase unzipping the DNA going the other way. So we're only looking at half this bubble here. And you kind of have to keep that in mind. So we're looking at this half of the bubble. Inside the bubble would be 2 replication forks, one here and one on the other end of the bubble. So DNA helicase comes in, and this does the unzipping of the DNA. And with this one, you have what's called gyrase or topoisomerase. That's this structure here, and it's outside the bubble. And this enzyme goes down the DNA with DNA helicase. And this enzyme keeps the DNA from getting tangled up. So it's always kind of unwinding the DNA as helicase is unzipping it. Now, what keeps the strands separated are called SSBPs. These are single-stranded binding proteins. So they attach to the actual backbone and keeps the DNA separated. And so there's your first part this is the initiation phase or step of DNA replication. Step 2 is elongation. So this is where it gets a little bit challenging. You have RNA primase. This is and I did it here with primary colors. So here's your RNA primase. RNA primase puts down RNA primer. So it's not DNA, it's actually RNA. And it puts down RNA primer. So it will have to get replaced in a later step. But for right now, RNA primase puts down RNA primer. So here's the primase, it's putting down the primer. It would be these 2 first and then this guy. And so it's putting down the primer, then the DNA can be laid down. So let's just look at this strand right here, which we'll call the leading strand, this bottom strand. So you got RNA primase puts down this RNA primer, and then right after that, along comes DNA polymerase. DNA polymerase puts down DNA, which is the goal here. This is what we need. So here comes DNA polymerase, and it's putting it down. And I'm laying it down in an order here so that you can see it puts it down in an order, and it's the 5 to 3' direction. And it's the only direction that DNA polymerase can put down DNA. And it's meaning it is built in the direction of, there's your 5 carbon right there, 5', there's your 3', so it's going to go, the arrow's pointing basically this way. So that means each nucleotide is put on 5 to 3'. So DNA polymerase comes along, puts down DNA 5 to 3 prime. The word anti-parallel comes up again because it's 5 to 3 prime on the other side, but it's going the opposite direction. So DNA polymerase comes in because the primer's down, and down goes our nucleotides, DNA nucleotides, in the direction of 5 to 3 prime. So this is the lagging strand, and it's going this direction. This is the leading strand because you put down one primer, the rest of your DNA can be built. So as long as helicase keeps going this way, the DNA on this strand is going to get built going that direction. This creates a problem for the other strand, and we're going to call this the lagging strand. Because DNA can only be built in the direction of 5 to 3 prime, that means you're going to end up with a bunch of little fragments that are going to be built, because helicase is going this way. So you're going to have another fragment here. So along at some point, a primer is going to be put down. Now, meaning RNA primer is going to be put down, DNA can be built going in that direction. But helicase keeps going, opening up more and more DNA. You have to put down a new primer. DNA is going to be built going that way. So that's why this is called the lagging strand. And it's got these little fragments, and these fragments we call Okazaki fragments. And they're going to be built just because it's fragmented as the DNA. This strand is just one the whole way. Put down one primer, DNA is built 5 to 3 prime. It's going in that direction. So we call this then the leading strand. And again, this would be the lagging strand that has these Okazaki fragments. And again, that's just due to the backbone of the DNA, and it can only be built 5 to 3 prime direction. And these new strands we're also going to call daughter strands. Now, after all that's laid down, now we have the termination phase. So in comes, so DNA polymerase is kind of done for now. Or not the primase, there's the polymerase. So in comes the endonuclease. Again, this is an enzyme that I've represented with basically a magnet here. So in comes endonuclease, or also called nuclease. This comes in and removes the RNA primers. So the RNA primers have been removed now. DNA polymerase can now put in DNA. So DNA polymerase comes in and lays down DNA, because that's what DNA polymerase does. And now all the other DNA strands are in. DNA polymerase can put that final one in. And this problem occurs again because it can only be built 5 to 3 prime. So endonuclease removes the RNA primer, DNA polymerase comes back into play, puts down DNA, and then DNA ligase comes in and actually attaches the backbone, connects these two together. Where the primer was located, DNA helicase comes in and attaches these, so now it's all connected. Now you got your 2 strands here, and you'd have your 2 strands here. This is called semi-conservative replication. What that means is the end products, you have a parent strand and you have a daughter strand when it's all said and done. Parent strand, and here's your daughter strand. So we call that semi-conservative replication. Because when it's all said and done, you got one original strand connected to, or opposite to the new strand. And that is DNA replication.